Today we will discuss a new bacilli that is named as Haemophilus, and it's a group, and the genus Haemophilus comprises a group of small non-motile, non-sporing, non-acid-fast bacilli, and they are gram-negative coco bacilli or rods, and often markedly pleomorphic and sometimes filamentous, that are parasitic on human beings or animals. So we are going to Haemophilus. And they are characterized by their requirement of one or more of two accessory growth factors, that are 10th and 5th, that are present in blood. And you all know there are so many factors in our blood, and out of them 10th and 5th factors are provided for them. And the name of genus comes from the requirement of these organisms for accessory growth factors found in blood, and that is heme means, and that heme comes from the Greek word that means blood. Okay, and philos means loving. So we can heme plus philos. Okay, heme means blood and philos means loving. We can divide it in two words. One is heme and second is philos. And this genus Haemophilus have species, and we have three species. One is Haemophilus influenzae, and second is Haemophilus ducreyi, and third one is Haemophilus afro. These are the three species, and Haemophilus influenzae is the species most commonly associated with diseases such as meningitis, epiglottitis, pneumonia, and septic arthritis, and localized disease of respiratory tracts including bronchitis and otitis media. These are the some diseases, and commonly associated with all these things. And when this Haemophilus ducreyi is well recognized as the etiological agent of the sexually transmitted disease, soft chancre or chancroid. And when we study the trachoma group, then we will discuss the same. Otherwise, you must know this is a species of this Haemophilus. And third one is Haemophilus afrophilus. Haemophilus afrophilus. And it is an uncommon but important cause of endocarditis. Okay, so these are the three species, and the other members of this genus are commonly isolated in clinical specimens, but they are very rarely pathogenic, and being responsible for primarily for opportunistic infections. And Haemophilus influenzae. Haemophilus influenzae is the first free living organism whose complete genome has been resequenced. So today we will study this Haemophilus influenzae. Okay. I have discussed that it has three species, and out of them this Haemophilus influenzae is important to discuss. And now we will discuss the morphology of this. And this is a very small gram-negative rods or cocobacilli, and the measurement of this 1.5 to 1.3 micrometer in breadth. And this exerting considerable pleomorphism, and I have already told that it is very changeable, okay, and it is non-motile, non-sporing, and non-acid fast. And in sputum, it usually occurs in clusters of cocobacillary form, while in cerebrospinal fluid, that is also known as CSF, from meningitis cases, usually appears cocobacillary, but bacillary and filamentous form and predominate. And cells from young cultures that is of 18 to 24 hours, it is a young culture, and are usually cocobacillary, while older cultures, इनको जो हम 24 घंटे से ज़्यादा देर रखते हैं, that are known as older cultures, are distinctly pleomorphic. Okay, earlier young cultures are cocobacillary, but old cultures are pleomorphic. Okay, मतलब आप 24 hours and more than 24 hours. Okay, and the virulent strains are often capsulated, which can be demonstrated by negative staining and Quellen reaction. Okay, I have discussed that this Quellen reaction earlier, so no need to repeat the same again. And the bacilli are relatively distinct or difficult to stain. And staining for 5 to 15 minutes with low flux methylene blue or dilute carbon fusion give good results. And this is all about the morphology, and now we will discuss about the cultural characteristics of the same. Cultural characteristics. And this Haemophilus influenzae is aerobic and facultative anaerobes, and it grows better in aerobic than in anaerobic conditions. It requires two accessory growth factors, as I have discussed earlier, that is 10th factor, and it is also known as hematine, and 10th factor that is found in blood, and it is also known as hemin. Okay, and the 5th factor, that is also known as nicotinamide, adenine, nicotinamide, adenine, and dinucleotide. Okay, factor 5. Factor 5th, that is nicotinamide, adenine, dinucleotide. Okay, and these accessory growth factors present in blood are essential for growth. And the optimum temperature for growth is 37 degrees centigrade, and growth is enhanced by a moist atmosphere supplemented with 5 to 10% carbon dioxide. And it cannot grow on nutrient agar as it lacks the accessory growth factors that are factor 10 and factor 51. Okay, so that cannot be grown on nutrient agar. So these need blood or chocolate agar. And the 10th factor, that is hemin and hemetin, is a heat stable, and it is required for the synthesis of the iron containing respiratory enzymes, that is cytochrome C. Cytochrome C. Okay, and cytochrome oxidase, peroxidase and catalase. And this 10th factor is not required for anaerobic growth. Okay, and again it is ये हीट स्टेबल है और इसके सिंथेसिस के लिए आपको आयरन कंटेनिंग रेस्पिरेटरी एंजाइम साइटोक्रोम सी, साइटोक्रोम ऑक्सीडेज एंड पेरोक्साइडेज एंड कैटालेज की जरूरत होती है. एंड नाउ द फैक्टर 5th, and this is a coenzyme nicotinamide adenine dinucleotide, that is also known as NAD, and in short NAD. Okay, and which acts as a hydrogen acceptor in the metabolism of the cell, and it is heat level being destroyed at 120 degrees centigrade in few minutes, and it is present in blood cells, and the growth is scanty on blood agar because only 5th factor is available in this blood agar. So 5th agar is present inside the RBC. So the chocolate agar is superior to plain blood agar for the growth of this Haemophilus influenzae, because 5th factor is released from within the erythrocytes into the medium and activates the serum amylase. Okay, na? These are the cultural characteristics. And the colonies. And if we talk about the colonies, the blood agar with Staph aureus streaks and chocolate agar are routinely used for the identification of this Haemophilus influenzae, and the colonies are small, translucent and non-hemolytic on blood agar. And capsulated stains produce larger, distinctive, iridescent colonies. And Field's agar is the best for primary isolation of this Haemophilus influenzae and gives a copious growth. Means ये बहुत अच्छी ग्रोथ यहाँ पे हमको मिलती है अगर हम फील्ड्स का एगर यूज करते हैं. फील्ड्स एगर. अदरवाइज ब्लड एंड चॉकलेट एगर आर गुड, बट अगर हम स्पेशलिटीज देखें तो ये फील्ड एगर इज ऑल्सो गुड, एंड इन दिस एगर वी गेट द कोपियस और प्रोफ्यूज और एबंडेंट colony or growth may be found. and the capsulated stage produce translucent colonies within a distinctive iridescent and Leventhals agar. So, in generally we use blood agar and chocolate agar, and these two fields agar is specific or special. Okay, so we need for growth of culture the different type of agars. Now we will talk about the biochemical reactions. Biochemical reactions. So again, this Haemophilus influenzae ferments glucose and galactose, what does not ferment sucrose, lactose and mannitol. And catalase and oxidase reactions are positive. Okay, and it reduces nitrates to nitrites, and it can be divided into 8 biotypes on the basis of indole production. And urease activity and ornithine decarboxylate reactions. Okay, these are the 3 things or reactions on which it can be divided into 8 biotypes. Biotype 1 to 3 are the most common, and biotype A1 is most frequently responsible for meningitis. Okay, and out of हम 8 बायोटाइप्स की बात की थी, उसमें से हमारे पहले 3 1 2 3 are the most common vacillized or co-vacillized for the responsible for different diseases, or in other words we can say these are the pathogens, and out of 3 biotype 1 is responsible or most responsible for meningitis. And while we talk about the resistance or viability of this influenzae, because it is a delicate organism and it is rarely killed by moist heat, that is at 55 degrees centigrade in 30 minutes. And if we refrigerate at 4 degrees centigrade or drying and disinfectants, so it can be killed by 55 degrees centigrade for 30 minutes, and if we do it refrigerate at 4 degrees centigrade and use disinfectant, it will be killed. And it also dies in 1 to 48 hours in dried secretions and airborne droplet liquid. And in culture, the cells die within 2 or 3 days due to the autolysis. Okay? ये अपने ही ऑटोलाइसिस की वजह से ये 2 से 3 दिनों में अपने आप ही ये किल हो जाता है. Okay? And now we will discuss about the pathogenesis that is produced by this Haemophilus influenzae. Pathogenesis. And as this Haemophilus influenzae is an obligate human parasite, and it resides principally in the upper respiratory tract, and it is non-capsulated stage of Haemophilus influenzae are regular commensal in the nasopharynx, okay, and throat, and especially in 25 to 80% of healthy people. माने हमारे 25 से 80% जो स्वस्थ व्यक्ति हैं उनके नाक में और गले में ये एक सामान्यतः पाए जाने वाला एक ऑर्गेनिज्म है ना, एंड इट इज नॉट naturally pathogenic for animals. What interpretational up to interperitoneal inoculation of large doses is fatal in mice. अगर हम किसी चूहा के पेट में इसको हम लगाते हैं या गिनी पिग में लगाते हैं या रैबिट्स में लगाते हैं तो इट इज कोई मतलब बीमारी पैदा नहीं करेगा. ओके, सो it is about the pathogenesis, and this causes invasive and non-invasive infections, and the invasive infections are caused by capsulate strains and non-invasive infections by non-capsulate strains, which are quite distinct in their epidemiological profiles. And when we talk about the invasive pathogenesis, invasive infections, one is meningitis, okay. Meningitis means the swelling especially in meninges, and it is the most serious of the disease occurring in children between 2 months to 3 years of age, and the case fatality rates is up to 90% in the untreated cases, okay, or maltreated cases, and majority of the cases are due to the type B strains. Okay, and second one is epiglottitis. And this is an acute infection of the epiglottis, which obstructive laryngitis, and it is especially seen in children above 2 years. And this condition is especially always associated with bacteria. ठीक है ना, इसमें हम अगर ब्लड का इन्वेस्टिगेट करेंगे तो वहाँ हमको बहुत ज़्यादा बैक्टीरिया दिखाई देंगे. ओके, एंड थर्ड वन इज निमोनिया. And this Haemophilus influenzae typically occurs in infants and is accompanied by emphysema and sometimes meningitis as well. Okay, इसमें emphysema हो सकता है और meningitis भी हो सकती है. And suppurative lesions, such as arthritis, endocarditis, and pericarditis may result from the infection of this Haemophilus influenzae. Okay, and next one is cellulitis. And it is seen particularly, especially in buccal and periorbital areas, and it is especially seen in young children. And I again repeat why this invasive infections we see that they are different type of infection. We see meningitis, epiglottitis, pneumonia, and suppurative lesions that are arthritis, endocarditis, and etc. And cellulitis, and these are the infections which are caused by this Haemophilus influenzae. And now we will talk about the non-invasive infections, in which we see that acute sinusitis, and otitis media. Okay, acute sinusitis and this otitis media are usually initiated by some viral infections. Okay, and there are acute exacerbations of chronic obstetric airways disease, COAD, chronic obstetric airways disease, and these are the acute exacerbations. एकदम अचानक अटैक आ जाना किसी चीज सांस में तकलीफ होगा, and they are initiated by the acute viral infections also. And this Haemophilus influenzae is an important pathogen associated with pneumococci in acute exacerbations of this chronic bronchitis and bronchiopatasis. Okay, so this is all about the pathogenesis, and in which we see that there are two types of infections: invasive infections and non-invasive infection, in which we see that different type of disease occur. Okay, and now we will talk about the laboratory diagnosis. Laboratory diagnosis. Okay, in which we like to have some specimen, and the specimen depend upon the type of lesion. So we collect a specimen like blood culture, for blood culture, and cerebrospinal fluid, throat swab, sputum, pus, aspirates from joints, middle ears and sinuses, etc. And from these sites we can have samples or specimens, and the collection and transport the same, and the specimens should be collected in sterile containers and under all aseptic conditions, and after collection we transport to the laboratory without delay, as these are the very sensitive or delicate organism, and it is very sensitive to the low temperatures, and therefore the clinical specimens should never be refrigerated at or below the temperature of 4 degrees centigrade. And after having the specimen, the pathologist do the direct microscopy by making a gram-stained smear, and especially in meningitis, gram-stained smears of cerebrospinal fluid shows pleomorphic gram-negative cocobacilli. Okay, तो जब भी हम ग्राम स्टेंड स्मीयर देखते हैं, ग्राम स्टेंड टेक्निक, उससे हम स्मीयर बनाते हैं, तो उसमें हम देखेंगे कि इसमें प्लियोमॉर्फिक ग्राम नेगेटिव कोकोबैसिली यानी डिफरेंट साइज के. ओके, एंड इम्यूनोफ्लोरेसेंट एंड क्वेलिंग रिएक्शंस जब जांच कराएंगे, इन्वेस्टिगेट करेंगे, तो दैट कैन बी एम्प्लॉयड बाय डायरेक्ट demonstration of this Haemophilus influenzae after mixing the specific type of antiserum. ठीक है ना, हम वो इन्वेस्टिगेट करते हैं, तो उसके लिए हम स्पेसिफिक टाइप ऑफ एंटीसीरम को यूज करते हुए ये करते हैं. एंड द एंटीजन डिटेक्शन कैन बी डन by testing the serum, urine, CSF or pus, and the technique used may be latex agglutination, coagulation, and counter-current immunofluorescence, and RIA and ELISA techniques. These are the tests which are done to find out this Haemophilus influenzae. And the culture, for CSF culture, and it should be plated promptly on the blood agar or chocolate agar. Okay, and that is not, we are not using for culture the nutrient agar, because हमने पहले बताया कि इसमें फैक्टर 10 और 5 की अवेलेबिलिटी नहीं होती, और हमें नाम से मालूम है हीमोफिलिस. ओके, ब्लड का लव करने वाला ये हमारा कोकोबैसिलस है. ओके, एंड ब्लड कल्चर, स्पर्म कल्चर ये हम कर रहे होते हैं. And for identification, the colonies of this Haemophilus influenzae are characteristic, and there will be a seminal odor, semen की तरह इसमें odor आएगी, smell आएगी, and confirmation of identity depends on the demonstration